Then that regulate our metabolic pathway. It's just like a tap, having a knob on top to control it. And without that, it will be very, very difficult for you to be able to regulate the water that flows in your house. And you know the headache that comes from damaged tap. So as a result of that, you try as much as possible to fix it. So the same thing applies to regulation enzymes, or the same thing applies to our metabolic pathway. They all need regulatory mechanisms which are actually carried out by enzymes that regulate the pathway. It is very important for you to note that there are different type of mechanism for regulating enzyme. We have the allosteric regulation, reversible covalent modification, proteolytic cleavage, feedback regulation, and regulation by isoenzymes. These are the five mechanisms by which different pathway different pathway used to regulate their in some metabolic pathway you can find this combination. It can be two, it can be three of the combination of mechanism of regulating the enzyme. Allosteric enzyme, this one is the most important and numerous among different type of enzymes that regulate our pathway. They have allosteric sites, and this allosteric site is different from the active site. This site often binds to small effector molecules that regulate the enzyme. These effector molecules can either be cofactor or coenzymes. Once they bind to this what they call it enzyme, they alter their conformation, their three-dimensional structure. They alter their active site, and this can be an either inhibitor or activator. And so when this happens, they affect the reaction. So inhibitor often slows down reaction, while activator often propels reaction forward. You can see subsequently that diagram before that when effector molecules bind to the allosteric site, which is distant from the active site, it does affect the conformational change, and it determines whether the enzyme will continue to act on the substrate or will not act on the substrate. Two perfect examples of allosteric enzyme are aspartate transcarbamylase and oxygen carrying protein hemoglobin. These are two perfect allosteric proteins. Cytidine triophosphate catalyzes the reaction of carbonyl phosphate plus aspartate, and leading to N-carbonyl aspartate. Then after 6 enzymatic reactions, we have cytidine triophosphate. And so once this enzyme this reaction produces sufficient cytidine triophosphate. The cytidine triophosphate, remember, it belongs to the pyrimidine family. It goes to inhibit the ATPase, and as it inhibits the ATPase at its allosteric site, remember allosteric sites are different from the active site, it stops further production of pyrimidines. Now this enzyme is very important for the balance between the pyrimidines and the purines. Now, the purines, when the pyrimidine pool is sufficient, cytidine inhibits the ATPase. Now, when the pyrimidine, when the pool of purine when the pool of purines are low, then ATPase goes to activate it for further production. So that's how this enzyme is very, very important. Remember, it makes use of allosteric form of regulation. That is, it can be activated by adenosine triphosphate or and inhibited by cytidine triphosphate. So this is very important for the balance between the nucleotide pool in the body. I realized that in your assignment, most of you did a lazy work, and you did not add up to what you have. Then the next form of regulation is reversible covalent modifications. Reversible covalent modification deals with formation of covalent bond, that's permanent bonds that are what reversible, that can be covalent bond are permanent bond, but in this case these bonds can be what can be reversed. And so there are different type of reversible covalent modifications that can take place. We have the methylation, hydroxylation, adenylation, and we have phosphorylation reaction. From their name, you can be able to factor the kind of reaction that takes place. Like phosphorylation is the addition of phosphates. Now, in some cases, the addition of phosphate group to a particular molecule actually activates it for to carry out its reaction. Then in some cases, the removal of phosphate group from a particular molecule actually inhibit it. So it can also be reversed. When you look at the examples, I've given you sufficient example which you can go through. You'll be able to understand the importance of this phosphorylation and this phosphorylation. So phosphorylation is usually catalyzed by protein kinase enzymes, and why this phosphorylation is usually catalyzed by phosphatase enzyme. So when you look at this, you'll be able to understand it. There are detailed information. In some cases, the phosphate group are added to the hydroxy part of the serine molecules. So in your notes, I've already explained and showed you how it takes place. A typical example, you have all these things in your glycolytic, most especially in your glycolytic cycle. We have exokinase, and we have some enzyme that have phosphatase enzyme. Another one is proteolytic activation of enzyme. This is another molecule, another means of regulation. We have I believe you understand what they call zymogen. Zymogen are pre-enzymes, enzymes that are produced in an inactive forms. So when they undergo proteolytic cleavages, these enzymes are split. Some molecules in them are being some compounds are being cut off, thereby activating them. These are the following examples that you have. And so your chemotrypsinogen, once it undergo proteolytic activation, it becomes chemotrypsin. We have your trypsinogen, we have your procollagen, procapsid, and so many examples. Even your prothrombin, they are being produced in the body, and when there's a cut, the interaction with ER and other cofactor makes it prothrombin. This is very, very important. These are the importance. It helps to prevent autocatalytic damage of the cellular components, assist in mobilization of enzymes in the cell, can be converted to active forms when they are needed, especially for what they call it these are very, very important means that the body used to regulate itself. We also have feedback regulation. This feedback regulation deals with the inhibition of the expression of the gene expression of this of this enzyme. Once a molecule is produced, unlike that of the allosteric regulation, this compound go directly and inhibit the expression, the gene expression. Stop coding for this enzyme, and immediately once it is being deregulated at the gene level, there is less synthesis of this enzyme. Thereby, at the end of the day, this the production of enzyme is reduced, then the reaction will stop being catalyzed. And lastly, we have regulation by isoenzyme. I've given you an example. I remember in the class I gave you an example of lactate dehydrogenase. In different various body parts, we have different lactase dehydrogenase, because they have different subunits, and different combination of subunits determines the type of what they call a lactate dehydrogenase in a particular section of the body, that is either in the liver, in the heart, or in the muscle. So if the body wants to regulate the lactate dehydrogenase, the regulation of lactate dehydrogenase in the body is localized. That is, it depends on the location, not it cannot be generally regulated. So these are the ways that the body used to regulate enzymes. So another important thing in the course, I also taught you about enzyme reconstitution. reconstitution. I remember I gave an example of transporting enzyme from from Lagos to Borno by land, and if it is in a liquid form, you expect there should be evaporation, and once there's evaporation, there will be more concentration, and so at the end of the day, it affects what you want to use the enzyme for. So the enzyme is being lysophilized. So for enzyme reconstitution, it's just making up an enzyme, returning it back to its original state, whereby it's able to obtain its three-dimensional structures and be able to catalyze the enzyme, what is able to catalyze its normal reactions. So reconstitution is the restoration of enzymatic activity by allowing the amino acid in the polypeptide chain of an enzyme to dictate its refolding and reassociation of the subunity in order to obtain its conformational structure for catalytic function. Is like once a protein is in powder form, it does not have its three-dimensional structure, it cannot catalyze reaction. But once you are able to reconstitute that enzyme, the amino acid dictates itself, it stands up and it half forms, and it's able to perform the the essential function. Now this is an important example. We have the porphyrin and the flavin groups. They are prosthetic groups that have various functions. And once this prosthetic group are removed from the once they are removed from the enzyme is unable to function, just like what you see in the picture. So reconstitution returns this prosthetic group back into the enzyme, and thereby the enzymes are able to perform their functions. So reconstitution efficiency depends on the initial agent of concentration, the type of agent used. The type of agent used also include the enzyme of interest, the composition of the lipids around the enzyme before your extraction, the rate of agent removal, and the ionic charges on top. These are the factors that depends on how successful your reconstitution is. Now, to remove membrane protein, you have to use detergent. Why? Because these proteins, some of these proteins are surrounded by cells, body cells, and these are phospholipids that hold the most of them may be integral proteins. Some of it are found deep into the into the cell. So they are surrounded by membranes. So as a result of this, you have to use detergent. This is not the homodic the Huawei or the Sunlight that you use. These are special detergents that we use in biochemical reaction, and what this detergent does is to solubilize the membrane for easy extraction of the for easy extraction of the protein of interest. So don't forget, it is very, very important that the type of protein you are using will determine the kind of detergent you are going to use. And there are different type of detergents that are being used. We have the ionic detergents, neutral detergents. They are different, but you can read it to see what they are being used for. We have zetronic detergents and the carrot-based detergents, and these are method of also getting your detergents out. When you use detergent, it is mixed with the protein, but you need to also extract your detergent from the protein of interest. So that's all about it. You can read, master the table, understand it, and you have the best. Now this leads us to reconstitution of lysophilized enzyme. I think I gave you an exam assignment on it. I saw people, they did a lazy job on it, but this is a typical answer for a reconstitution of lysophilized enzyme. Lysophilized enzyme have powdery form of enzymes. So this aspect is very, very simple. Just read through it and you'll be able to understand how they reconstitute lysophilized enzymes. So lysophilized enzymes are carried out because to avoid a whole lot of things: denaturation, concentration, over concentration, and so many things. That's why your enzyme is lysophilized before traveling. And when they lysophilize it before traveling, they also give instructions of how to reconstitute it so that the enzyme can best function properly, and also so that you can be able to achieve your interest, your the purpose why you secure the enzymes. So I think that's all about the course. I wish you the best in exam.